Alright guys, let's talk RNA. So there's going to be some differences between RNA and DNA. In the lab you did on DNA replication, at the very end, the last section of that lab looked a little bit at RNA, so you might know a little bit about this already. But I just wanted to do a quick screencast and run you through some of the major differences. First of all, in DNA we know we have 4 different bases, and those bases are A, T, C, and G. Alright, so those are the bases, those are DNA's bases. So I'm just going to write that up here real quick, if you can read that hopefully. DNA's bases. Alright, so those are DNA's bases, but it's different. In RNA, we replace one of those bases. So in RNA, we have 4 bases. We have adenine, so we have A. We have cytosine. We have guanine, and we know that guanine always goes with cytosine. And we also have uracil. So basically in RNA, what's happened is we have replaced the thymine base with uracil. So now A goes to U. So wherever we would have a T in DNA, we now put a U. So that's one of the main differences in RNA. The other big difference in RNA is that if I'm going to just draw like a real quick RNA molecule, I'm going to draw it like this because RNA is single-stranded. All right, so it's a single-stranded piece of piece of nucleic acid. It is RNA, one strand. It has 4 bases, an A, a U, a C, and a G. And then another big thing is that we're going to replace the sugar. So in DNA we have a deoxyribose sugar. In RNA we have a ribose sugar. So let's take a closer look at RNA real quick. So here we go, guys. I have a picture of DNA here and I have a picture of RNA here. We're going to look at the differences real quick with the pictures, and we're going to try and see if we can understand what happened. And there's actually, as far as I'm concerned, a mistake here, and we'll point that out in a second. Quick review. DNA. What do we have here? We have 2 strands, strand 1, strand 2, bound together. Remember that the phosphate here and the sugar here make up our rungs on the side of a ladder, and that our 2 bases joined together or bonded together make up our rungs. So if we were going to climb the ladder, we'd climb up the bases, and the stuff on the side would be the phosphates and the sugars. Perfect. So what's the big deal? What's different here? Well, first of all, I can tell you this. If we're looking at deoxyribonucleic acid DNA, deoxyribonucleic acid is called that because it's made of a deoxyribose sugar. And that deoxyribose sugar is right there. But in RNA, that's different. It has a ribose, that's an R, sugar. I've heard you complaining about my writing, and that's right there. So in RNA, ribose sugar, so it's RNA, R for ribose. Deoxyribose is a deoxyribose sugar, D for DNA, and then, or D for deoxyribose sugar. So then, what else is different here? Well, this is double-stranded. So this is 2 strands. This is a single-stranded piece of nucleic acid. And then the last major difference that we're going to talk about here is this little guy right here, the uracil. In RNA, like we said in the last slide, we replace all the T's with U's. Now there's a mistake here, and let's see if we can find it. I'm going to point, whoa, that's a big pointer. I didn't want that. I want this little guy. I'm going to point here at this A. So I want you to picture that this piece of DNA splits right down the middle, just for a short time. And this is what you're getting ready to learn about. This is how we make RNA. It splits for a little time, and then the RNA free nucleotides. And think back to what a nucleotide is. A nucleotide is this phosphate, this sugar, and its base all together. Phosphate, sugar, base. Those are all nucleotides. So there's these free nucleotides just floating around in the nucleus. And they come up and they match up with these things. So if I have an A right here, it's going to match up with the corresponding base in RNA, which is a U, because A's go to U's in RNA. There is no T. The T is replaced with a U. The G right here is going to match up with a C. The C is going to match up with a G, and here's the mistake. The T is going to match up with, oh no, not a C. It's going to match up with an A. So that would be an A. So those are the major differences. Single-stranded, the ribose sugar, and we get rid of thymine and we replace it with uracil. So we have U's now instead of T's. Okay, so to end this screencast, I quickly wanted to talk about the 3 types of RNA. And I have these 3 pictures here that hopefully are going to help you remember what they are. The first type of RNA, and the one that we're going to talk about the most, is mRNA. This is messenger. That says messenger, guys. Can you read it? Messenger RNA. And what messenger RNA does is it takes a message from inside the nucleus and takes it out to the rest of the cell. This is how DNA communicates the code it has to tell the cell what to do. So the DNA will split apart, we'll make a piece of RNA. That RNA will leave the nucleus, go out to the cell, and it sends a message there. Well, when we send a message there, we need to know what the message says. Okay, so the next type of RNA we're going to encounter is the rRNA. Now you might be asking yourself, okay, I get the bird from messenger RNA. It's carrying a message from the nucleus out to the cell. But what's the pirate? I don't know. rRNA, right? It's I call it the pirate DNA. It has nothing to do with pirates, but maybe it will help you remember rRNA. This is the ribosomal RNA. All right, so this is the ribosomal RNA. That's the messenger RNA. I can stick that there if you want. So what the ribosomal RNA does is really, together with a protein, it forms together to make a ribosome. So that messenger RNA is going to leave the nucleus, go out to the cell. It's going to hook up with a piece of rRNA and a ribosome, and now we need to figure out what it says. Well, when we need to figure out what things say in class, sometimes what do? We go to a translator. A lot of students are using that translator in Google Docs to translate their documents from English into Spanish or something. Same kind of thing. And that's the final type, is the tRNA. And this is the transfer RNA. Now think about the picture. What's happening? We're taking files and we're transferring from the computer up to the internet, or from the phone to the internet, or we're going from the internet down to the phone. So we're transferring files. And transfer RNA, what it does is it figures out how to translate the message we send out by transferring the pieces the message calls for. I'm not going to go any deeper than that because you're going to learn about that later. But I thought it was a good point to talk about this. There's 3 types of RNA: mRNA, rRNA, and tRNA. Messages, ribosomal, transfer. Where do they come from? They come from the cell's nucleus, where the DNA splits apart and we make a piece of RNA that's single-stranded. Let's do this. That we make a piece of RNA that is single-stranded, has uracil instead of thymine. So we're going to put over here in parentheses, no T's, right? No thymine. And it has a ribose sugar. Great. Hey, see you next time.